Well, so let's start with the urinary system. So we are also going to be talking about the urinary system in the lab today. So the organs that form part of the urinary system are the paired kidneys, the tubes that emerge from the kidneys, which are called the ureters, that transport the urine to the bag-like structure called the urinary bladder, which is just behind the pubic symphysis. It's behind the pubic symphysis. And then you have the passage through which urine is voided. This is called the urethra. So don't get confused between ureter and urethra. Okay? Urethra is the passage from the urinary bladder to the outside. The urethra is shorter in females, it is longer in males as it has to pass through the penile tissue. So that's the reason why urinary tract infections are more common in women than men, because the urethra is short, so the bacteria from the external genital region can easily enter into the urethra, the bladder, and then into the ureter. So they can urinary tract infections are more common. Now the bladder, the paired bladders are situated high up in the abdominal cavity, okay, behind your floating ribs. And if you look at the layout, so each urinary bladder is like each kidney is like a bean-shaped structure, maybe about 8 to 10 centimeters in length. And the right kidney is always a little lower than the left kidney. Okay? The reason for this is because we have a huge organ here that pushes the right kidney down, and that is the liver. So the liver is a solid, large organ in the abdominal cavity. So the right kidney is a little lower than the left kidney. Now, when you look at the location of the kidney, we use the term for location of the kidney we always say retroperitoneal location behind the parietal peritoneum. Okay, so what is retro means behind the peritoneum. So what does this actually mean? So if you were to look at the abdominal region of the body from the side, okay, when you look at a lateral view, you have the vertebral column. Okay, I'm just diagrammatically drawing it so that you understand. So this is your vertebral column. That's the diaphragm. Okay. The kidney is located just on top of the vertebral column. Okay, so way behind, it's not in front. So if you were to cut the abdominal wall, you will not see the kidney. You will see the liver, you will see the stomach, the intestines. So the kidney is way back. Okay, so this is where the kidney is located. And let's say this is the anterior abdominal wall. This is the anterior abdominal wall. Just like the pleura, the pericardium, which is made up of 2 layers, the abdominal cavity is also covered by membrane called the peritoneum. So you have one layer of the peritoneum which is covering the inside of your abdominal wall, like that. Okay. We call this the parietal peritoneum. So it's a thin membrane which lines the inside of your abdominal cavity, and the parietal peritoneum is going to go like that. Going to form a bag like that. And inside this bag you will have, the liver is covered by this, the stomach is covered within this bag, the intestines are within this bag, okay? And they are in turn covered by another layer of the peritoneum. Each organ is covered by another layer of the peritoneum. This is called the visceral peritoneum. Visceral peritoneum. So just like the pleura, which is the parietal pleura covers your inside of the chest cavity, the visceral pleura covers the organ, the lungs, the same way the peritoneum is also the same. The parietal peritoneum is like a bag, okay, and then it's a double layered membrane, so, and then it kind of folds and then covers each organ, okay. But the kidney is not covered by the peritoneum. It is behind the parietal peritoneum, it's behind the bag, okay. So the organs that are not covered by peritoneum. So there are a few organs that are not covered by the peritoneum. One is kidneys, pancreas, and rectum. Kidney, pancreas, rectum are not covered by the peritoneum. All other organs are covered, yes. Okay. And also, this is another reason, location is another thing. Another reason why any problem in the kidney always results in back pain, because it's close to the vertebral column. So low back pain, that is a sign of something going on in the kidney or in the urinary system. Okay, so important to know some of these connections. Okay, now although the kidney is behind the parietal peritoneum, it's not covered by the membrane. The kidney is protected, okay? So there is a thick connective tissue that covers the kidney, that is called the renal fascia, thick fibrous connective tissue, and then it is also surrounded by fat, renal fat, which protects the kidney from any damage, okay? And on top of each kidney, you have the adrenal gland. Okay, you have the adrenal gland sitting like a cap on top of each kidney, and you know one of the hormones of the adrenal cortex is aldosterone, and aldosterone affects retention of sodium. So the hormone is secreted by the gland which is very close to the kidney. Okay. Now let's look at so that's how it is, okay, right at the back. What are the functions of the kidney? We do know the obvious function is to remove waste products. Okay, that's an obvious function of the kidney. All of us know that. What are the waste products? Urea. Urea is a byproduct of protein breakdown. Okay, now these are things you should know. Where does urea come from? Protein breakdown. Okay, uric acid, a breakdown of DNA and RNA, whatever you eat, right? DNA is there in your animal protein, vegetable protein, so.
Breakdown of protein?
Protein, yes, protein breakdown. Uric acid is breakdown of DNA and RNA leads to uric acid. So uric acid, also when you eat certain specific vegetables and fruits, it will have uric acid in it. So uric acid is a throwaway product. Creatinine. Now, creatinine is the breakdown product of muscle creatine. your muscle, especially skeletal muscle, has this protein called creatine phosphate. Creatine phosphate, which allows for quick bursts of ATP. So muscle stores creatine phosphate so that if you're running a 100-meter sprint, the ATP comes from the breakdown of creatine phosphate. So this is a high energy bond. So when you did skeletal muscle, you would have learned about this. So when creatine breaks down, you get creatinine. And creatinine cannot accumulate in the body, it has to be thrown out. Bilirubin. Bilirubin comes from breakdown of heme. So that also needs to be thrown out. So that's the reason why your urine is yellow in color. Pale yellow because of bilirubin, which is a yellow colored pigment. So, and any other waste products, acids, alkalis, which comes from your diet, all of that has to be removed. So water, extra water, excess salt, all of that has to be thrown out. So those are obvious, this is an obvious function of the kidney. But apart from that, the kidney has innumerable functions which are very important. So if the kidney is damaged, all of this will be affected. Water balance, okay? Water balance will be affected. Electrolyte balance, electrolyte sodium, potassium, calcium, okay, will be definitely affected if the kidney is not functioning properly. pH of blood, because the kidney is the one that throws out extra acid from your body, throws out extra alkali. So it maintains the pH of the blood. So the moment the kidney is not functioning properly, you will have changes in the pH of body fluids. Blood volume and blood pressure, we have already seen that, renin, angiotensin, aldosterone mechanism, very important. So one reason for high blood pressure is kidney disease. Okay, so whenever somebody goes with high blood pressure, they will check kidney functions, because if the kidney is damaged, blood pressure can be high. Okay, it is not going to function properly. Red blood cell production, hormone erythropoietin is also released from the kidney. Okay, regulating calcium levels, parathyroid hormone. Okay, affects the kidney to form active vitamin D. So all of this, you can see lots of important functions. This is not the only function. So if the kidney is damaged, it can lead to multiple system disorders. There could be a lot of changes. Now, this is something you will be seeing in the lab today. You will be looking at these models. So it's going to make it easier. You can just go straight ahead and start looking at the model and identifying these parts. So let's do that. Okay, let me see. When you take the kidney and you slice it lengthwise, this is a longitudinal section. Okay. So these are the parts you should be able to identify on a kidney model. So lengthwise section, the outermost covering of the kidney is called the capsule. So for everything we use renal as a prefix to indicate that we are talking about the kidney. Renal capsule, this is a fibrous outer layer. Okay, and then you are going to see the ureter emerging from the medial aspect of the kidney. So this is lateral, this is medial. So from the medial aspect of the kidney, you're going to see the ureter. At the same point, you're going to see the renal artery bringing blood into the kidney, and the renal vein carrying blood out. So this central region, you're going to see the artery, the vein, and the ureter coming out. Wherever blood vessels enter and leave an organ, that region is called the hilum. So the hilum is a very common term. It's a common term for the area where blood vessels enter an organ and leave an organ. So this is the renal hilum. This region is the renal hilum. Okay? When you slice the kidney, you're going to see that the outermost region, this outer region, okay, is a little lighter in color. You can see it even in a normal kidney. This region is lighter. And since it is outside, we call this the renal cortex, just like adrenal cortex. So cortex is again a very general term meaning the outer region of an organ. Okay, renal cortex. The inner region is more darker. Okay, it's darker simply because you're going to see some triangular shaped structures. About 7 to 8 in each kidney. Okay, you're going to see these triangular structures, and these are darker in color. This inner region, this inside region is called medulla. Just like adrenal medulla, renal medulla. So outside is renal cortex, inside is renal medulla. Okay? So all of this is there in your lab book. You are going to be seeing this on a model. You have to identify all of this. Yes. 7 to 8 in each kidney. Each of this triangle is called a pyramid. Renal pyramid. These are the ones which are going to be like darker in color. Okay? Cortex, these are the pyramids. Now the cortex, sort of this light, these are in between the pyramids, it's still kind of lighter, the same color as the cortex. So these in between regions that you see here, these are called renal columns. These are called renal columns. Okay? At the tip of each pyramid, from each pyramid, you're going to see a small tube emerging. Okay, I'm just drawing these three. You're going to see a small tube emerging. And then about 2 or 3 of them may join together to form a bigger tube. So let's say there's another tube here, and then these 2 will join together to form a bigger tube. So here's one, here's the other one. Now each of this smaller tube that is at the tip of the pyramid, this is called minor calyx. Minor calyx. And when they join together to form this bigger tube, this one here, this is called major calyx. And so there will be tubes from here as well. Okay. And all these major calyces, remember these are just tubes, pathways. All these major calyces will open into this funnel-shaped region, which is the starting point of the ureter. So this region here. This broad funnel-shaped region, that is called the renal pelvis. Renal pelvis. Okay. So once again, outer region, renal cortex, inside renal medulla. In the renal medulla, you're going to see these triangles, dark colored triangles, which are called renal pyramids. From each pyramid, there's going to be a small tube that emerges. that is a minor calyx. 2 or 3 of the minor calyces will join to form a major calyx, a bigger tube, and all of them will ultimately open into this funnel-shaped starting point of the ureter, which is called the renal pelvis. Okay, this tube is the renal pelvis. This will continue down as the ureter. And the pale regions in between the pyramids are called renal columns. Okay, they are called renal columns. Now, if you look at it microscopically, the tip of each pyramid, at the tip of each pyramid, you're going to see lots of tiny microscopic holes. There are lots of tiny holes here. Not visible with the naked eye, but under the microscope. Tiny openings. Now, these tiny openings that are seen at the tip of each pyramid are called renal papillae. They are called renal papillae. Okay? This is the gross look of the kidney. So, if you slice up, you're going to be able to see this. But microscopically, what is there in the cortex? What is there in the medulla? What are those tiny holes? How is urine formed? That's the next thing. Histology. What is it that is there in the cortex and in the medulla? So, what is a cell that gives this structure to the kidney? So, what's there in this tissue? So, the basic structural and functional unit of the kidney is the nephron. So, just like your nervous system is made up of neurons, the structure that is actually filling up the kidney and also responsible for the function of the kidney is the nephron. Okay? Now, the nephrons are microscopic structures. So, you're not going to be able to see them unless you take a piece of tissue and look at it under the microscope. Now, these nephrons, how many are there? Each kidney has about 1 million, roughly, okay? Roughly, approximately 1 million nephrons. That means we do have 2 million in total. But our bodily functions can be, taken care of with just 1 million nephrons. We don't need 2 million. 1 million is enough to take care of all the functions that we looked at earlier. Okay? That is the reason why kidney donors or kidney transplant is one of the first organ transplants that was done, because a person can survive with just one kidney. One kidney is sufficient to maintain blood pressure, blood volume, electrolyte, water, acid base, get rid of stuff. Okay? So, that's the reason why people, they can donate a kidney to someone who needs it, who doesn't have both the kidneys functioning. They can donate it. Okay? So, about roughly 1 million nephron in each kidney. Okay? Now, where are these nephrons located? Out of this 1 million in each kidney, 80 to 85%, that is 800,000 to 850,000 nephrons, are located in the cortex. They are located in the cortex, that is this outer region. They are located here. Okay? We call them cortical nephrons. Because they are in the cortex, we call them cortical nephrons, which are responsible for almost all the functions that we listed at the beginning. This is what they do. So the remaining 15 to 20%. So 80 to 85 is in the cortex. The remaining 15 to 20 is actually close to the pyramids. Okay, they are close to the medulla. They are not in the pyramids, but they are close to it. Okay, so this 15 to 20% located close to the medulla. We call these nephrons juxta. Juxta means near, close. Juxtamedullary nephrons. Juxtamedullary nephrons. And the juxtamedullary nephrons have a very important role in one of the functions of the kidney, water balance. So they have an important role to play in water balance.
You said one of them?
One of the functions, I said, water balance. All functions are subserved by cortical nephrons, whatever we saw. Water balance, electrolyte balance, pH, blood volume, blood pressure. Cortical nephrons do most of that. The juxtamedullary nephrons are specifically geared to maintain water balance in the body. Okay? So now we look at the structure of a nephron. So what is the structure of a nephron? So we'll talk about blood vessels. Structure of a nephron. So this is the second thing. So you will be looking at the cross section of the kidney, longitudinal section of the kidney. In the lab, we have models, very good models, where you can see all of that we listed. Now, the second model that you will be looking at is a nephron structure. Okay. Now, lots of new terms here, anatomical terms. So try and stay with me and follow this, and then when you see it in the lab, it'll make it a little easier. Okay. So let's draw the nephron. And of course, all of these pictures are there in your book, so you can always, if you're the type who likes to just look at the picture and study, that's fine. But, I'm still going to draw it because, when you draw something yourself, it just sticks in your memory better, okay? Rather than just looking at a picture, if you draw it, then it stays. So we are talking about individual nephrons. So we have roughly 1 million like this, okay? A nephron is made up of two parts. Each nephron is made up of two parts. The starting portion of the nephron, the first part of the nephron, is called the renal corpuscle. Renal corpuscle, the C is silent, okay, renal corpuscle. The second part of the nephron is called the renal tubule, because it's a microscopic tube, so it's called a tubule. Okay, so this is the starting point, renal corpuscle, and then a renal tubule. Okay, now let's see how it looks like. Yes. Okay, so let's look at the renal corpuscle. The renal corpuscle is made up of two parts. Okay, this in turn is made up of two parts. Renal tubule is made up of three parts. So the renal corpuscle, what is it made up of? It is made up of a structure called glomerulus, and glomerular capsule, or the older term which many physicians will still use, if they're teaching this, Bowman's capsule. Bowman's capsule, to honor the person, but the more recent term is glomerular capsule. So what is glomerulus? Glomerulus is just capillaries. Okay? Glomerulus is a network of capillaries. It's a network of capillaries like that. Okay? Blood enters the glomerulus. Okay? Blood is brought into the glomerulus through an afferent arteriole. So now you already know arteriole and all that. So arteriole brings the blood in, and then blood leaves the glomerulus through another arteriole called efferent arteriole. Afferent means going towards an organ, efferent is going away from the organ. If you remember nervous system, sensory nerves which carry information to your brain are called afferent nerves. Motor nerves which bring the information from the brain to your muscles and glands are called efferent nerves. So afferent and efferent are common terms. Afferent brings the blood in, efferent takes the blood out, and this is the glomerulus, the capillaries. So here is a structure which is slightly different from what you learnt in cardiovascular system. An arteriole enters the organ, breaks up into capillaries, and after capillaries, what is the blood vessel? The venule. Whereas here, it goes into another arteriole. Okay, so that's one big difference that you see in the kidney. An arteriole feeds blood into the capillaries, and another arteriole carries the blood out. Okay, so the kidneys are said to be, if you look at the kidneys, dual capillary network. We will see later where this dual capillary comes from. So two sets of capillaries. The first one is the glomerulus. Okay, so don't get confused with that now. So this is the glomerulus. And where is this capillary sitting? It is sitting inside a cup. It's sitting inside a double-walled cup like that. So the capillaries, so if these are the capillaries, like if you have a punching bag and then you punch into that bag, so the bag is going to cover your fist. That is exactly how the glomerulus and the glomerular capsule is. So the capillaries are your fist, and that punching bag that covers will be your capsule. So it's a double-walled capsule. That means there is a space here. So you see I've drawn it like that. So the capillaries and then the cup. So this is the glomerular capsule. This is the glomerular capsule. This is the first part of the nephron, glomerulus and glomerular capsule. Together you call it renal corpuscle. What happens in this place? It is in the renal corpuscle that filtration of blood occurs. Filtration of blood occurs here. Okay? You can also think of it like your coffee pot, the coffee maker. So the top where you put the coffee grounds in, that is like your capillaries. And then the pot in which you collect your coffee, that is your glomerular capsule. Okay? The only thing here, the glomerulus is covered by the capsule. Okay? It's like a cup. And it's a double-walled cup, so there is a space in between. There's a space in between where the liquid can collect. Now comes the second part of the nephron, which is the tube. From the cup, the tube that emerges is quite twisted. It's a twisted tube, coiled tube. It's not a straight tube. This is called proximal convoluted tubule. Convoluted means twisted or coiled. Proximal, why? Because it's close to the cup. So it's called proximal convoluted tubule, PCT. So renal tubule, the first part is PCT. Then the tube straightens out. It straightens out and it dips down like that. It goes down like that. Yeah. It dips down like that and then goes back up again. Goes back up again. Okay? So think of it, look at this structure. So the starting point of the nephron will be somewhere in the cortex, and then the tube will go down like that and come back up again. Okay? It will go all the way down to the border of the cortex and the medulla, or sometimes even enter the medulla and then come out. Okay? So this second part, so this is the first part of the tube. The second part of the tube is called the nephron loop. This is called the nephron loop. Okay? This is the descending limb because it's going down. Descending limb going down like that. And this is the ascending limb of the nephron loop. Ascending limb. So the fluid will flow down like that and then go up again. Okay? It's also known as, the older term is loop of Henle. Loop of Henle is the older term. The newer term is nephron loop. And then again, as it approaches the same nephron, okay, you see that ascending limb is going to come very close to the starting point of the nephron. And when it comes there, it again becomes twisted and coiled. It again becomes twisted and coiled. This is the third part of the tube. This is called the distal convoluted tubule, DCT. The distal convoluted tubule. And this distal convoluted tubule has to open into something. It opens into a bigger tube. This is called the collecting duct. This is called the collecting duct. The collecting duct is not part of the nephron structure. Okay? If you are asked what is the structure of a nephron, it is made up of two parts, renal corpuscle, renal tubule. Renal corpuscle is made up of two entities, glomerulus and glomerular capsule. Renal tubule has three parts, proximal convoluted tubule, nephron loop, distal convoluted tubule. Imagine there are 800 to 850,000 nephrons in the cortex. Okay? So all of them have this structure, and then yes, I wanted to show you this picture. So many nephrons, thousands of nephrons, the distal convoluted tubule will open into the collecting duct. Okay, so there are 1 million nephrons. So you don't have 1 million collecting ducts. Many nephrons will, the distal convoluted tubule that I showed, like opening into the collecting duct, this is one nephron. And then there will be multiple ones. which will open.
And that's what ADH works on?
Yes, this is where collecting duct ADH works on this. And this collecting duct opens at the tip of the renal pyramid. And this is what I told you, the tip of the pyramid has lots of tiny holes. What are those holes? They are the openings of the collecting duct. This opening is called renal papilla. Renal papilla. So you see here, these are the cortical nephrons, the 80 to 85% which are at the top. Their loop will dip into the pyramid a little bit and then go back up. This is the juxtamedullary nephrons, which are the starting point is very close to the medullary region. This is the cortex, this is the medulla. So their starting point is closer to the medulla. That's why you call it juxtamedullary. And then if you see, they have much longer loops which go all the way down into the medulla and come back up. Okay? So that's how the only difference between the cortical nephron and the juxtamedullary nephron, these nephrons have longer loops, whereas the other one has shorter loops. They have much longer loops, which allows them to maintain water balance. Okay, that's why they have long loops like that. Okay, now what each nephron is going to do all these functions that we talked about. So the first part of the nephron, where you have the cup and the blood vessels, that is where filtration occurs. So things get filtered out. So as blood flows through these capillaries, things get filtered out. Okay, we'll talk about what gets filtered out. So because you want to remove the waste products. So things get filtered out. And this filtrate, what comes out into this cup is called the filtrate. This is called the filtrate. Do not call it urine. If you say that the renal corpuscle filters urine, that means you have not studied A&P. Okay? It's not called urine. It is called filtrate. Okay? Now this filtrate then moves down this tube. Okay? It goes down this tube. And as it goes through this renal tubule, a lot of changes are going to take place to the filtrate. Many changes. We'll see what happens. A lot of changes take place. And then it goes through PCT, descending limb, ascending limb, DCT, collecting duct. The moment the liquid emerges from this papilla, that is the point you call it urine. Because after that, no change is going to take place. It is just going to flow out of your body. So let's go back to this. So the tip of the pyramid is where you have the opening of the collecting duct. Up until that point, a lot of changes are going to happen to whatever has been filtered. The moment the filtrate comes out of the collecting duct into the minor calyx, it is called urine. Because after that, it is just going to flow through the minor calyx, major calyx, pelvis, ureter, urinary bladder, urethra, and out. Okay? So at this point, when the liquid comes out into the minor calyx, that's when you call it urine. Okay? So we could do this, and this will be helpful to you. So we can write down the pathway of the filtrate and urine. So pathway of filtrate and urine. Okay. And once you sort of study like this, you will not forget it also afterwards. So where does the filtrate start? Filtration occurs in the first part. So we will start with filtrate in glomerular capsule. Filtrate in glomerular capsule. This is where it is starting. From here, the filtrate is going to go into the proximal convoluted tubule. That's the next part. So we can go here. From the proximal convoluted tubule, the filtrate is going to enter descending limb of nephron loop. Next, it is going to go into the ascending limb of nephron loop. Next, distal convoluted tubule. Next, collecting duct. Okay. From the collecting duct, renal papilla. That is the small tiny opening. Okay. That's it. So up until this you call it filtrate. Now it will be urine, flow of urine after this. So this one, filtrate. Okay, you can never use the word urine because you'll be wrong. From the renal papilla, it is going to go into the minor calyx, major calyx, renal pelvis. I'm just going through all the anatomical structures. It's all a continuous tube. Renal pelvis, ureter, urinary bladder, urethra, and out. Okay, so this is the pathway of filtrate and urine flow. Okay. So filtration occurs at the first part, and then in the tube part there are lots of adjustments going to happen. Yes. Okay, so that is nephron structure. Now we have to look at this part where filtration is occurring. How is it that blood you're able to filter? Okay, what is the structure of this cup? So here you just have the picture of the cup. This is the third model that you'll be looking at. So the first model is this longitudinal section of the kidney. The second model is the nephron structure, where you will be able to identify every part. And then the third model is this. Okay, structure of the cup itself. So let's do it here. So instead of drawing this whole cup like that, we'll do it this way. So let's draw the Bowman's cup. It's a double-walled cup, so there is a little space where the liquid can collect. Okay, so this is the glomerular capsule. Glomerular capsule. Now the glomerular capsule is lined by, it is a structure that is lined by a single layer of flat cells, so simple squamous epithelium. Okay, simple squamous epithelium lines the this one. This is simple squamous epithelium. This is the lumen, the space, lumen of the cup. This is the lumen. Okay. Now this outer wall of the cup, this outer wall, which is lined by the simple squamous epithelium, this is called the parietal layer of the Bowman's capsule. Parietal layer of the glomerular capsule. You know, parietal is always outside, visceral is inside. So this is called the parietal layer of the glomerular capsule. Okay, so you can see here, it's just one layer of flat cells. Now this inside layer, this inner wall of the cup, here also it is still simple squamous epithelium. It is still simple squamous epithelium, but the cells are modified. How are they modified? They have a lot of openings. Okay, they have like foot processes like these, and there are lots of openings for things to filter out. Okay, so this inner wall, which is called the visceral layer, has modified squamous epithelial cells, which are suitable for filtration because you want things to come into this cup. Now, these modified squamous epithelial cells are called podocytes. These are called podocytes. Podocyte means foot processes. So, these extensions that you see, that's why it's called a podocyte. So things can easily filter out from here through all. So it's fenestrated. Fenestrated means there's a lot of tiny openings. Okay, so that's the visceral layer, and then you have the blood vessel. I'm only going to draw just one blood vessel. This is the afferent arteriole bringing blood in. This is the glomerulus, which is almost stuck to the inside wall of the cup like that. And then this one is the efferent arteriole, blood goes out. This is the glomerulus. Now you know that capillaries are also lined by a single layer of flat cells, endothelium. So what filters out, it's very easy. So here you have blood flowing. You have blood flowing through the glomerulus. So when things have to filter out, they don't have to go through a thick membrane. They just have to cross this endothelium, which also has a lot of holes in it. So it has to cross this. It has to cross this fused basement membrane. And then it has to cross this podocyte, and it has to get into this cup. Okay, so filtration is an easy process because the membrane itself is not very thick. Okay, it's not very thick. So you see this inside this podocyte, this yellow structure is the podocytes. So the podocytes are almost stuck to the capillaries like a cling wrap. So they are stuck to them. So it's very difficult to peel them out. Okay. So this is the outer layer, this is the inner layer, which is almost stuck to the cup. For example, if you are the same punching bag example, if you're punching your fist into a punching bag, there is the bag covering your fist, and then there is air inside the bag and then the outer wall of the bag. So that part of the bag that is covering your fist is the podocytes. So they are almost stuck there. Okay, they are kind of stuck. And then the outer layer of the bag is the parietal layer. Okay, so that's how this is. And it is best suited for things to come out easily. So blood flows in through the afferent arteriole, blood flows out through the efferent arteriole. Now this picture is very, it shows you exactly how the afferent and the efferent arteriole are. The afferent arteriole is much wider than the efferent arteriole. Okay, it's a much bigger blood vessel than the efferent. Okay, and there's a physiological importance of that. So this arteriole is a little bit more bigger than this one. Now comes what is it that filters out? When we look at things are as blood flows through these capillaries, I told you the filtrate will collect here. So what does the filtrate have? Now anything that is tiny, small molecule, uncharged, no positive or negative charges, will easily filter out. Anything that is tiny will come out. Okay? Anything that is big will be prevented from coming out through those openings. So what is the composition of the filtrate? Composition of filtrate. Obviously, the first thing is water. Water being a small molecule, water will come out. Any small molecules that are dissolved in the plasma, okay, because this is blood, so water from the plasma is going to come out. All small molecules, okay, salt, sodium, although they are charged, sodium, chloride, calcium, potassium, these will all come out along with the water. Glucose, tiny molecules, glucose, amino acids, fatty acids, anything that is fat soluble can also easily come out. Uncharged, but these being tinier, they can come out easily. Okay, the size. So size is one and charge is another. If it's a larger molecule with a charge, it will not come out. So we'll see what won't come out. All the waste products, okay, waste like urea. Urea is fat soluble, so it can come out. Uric acid, creatinine, bilirubin, okay, acids. All of this will come out in the filtrate. Okay. What will not come out? Things that cannot be filtered. Most important, plasma proteins, they cannot come out. Even albumin is big, so it cannot come out through those tiny openings. Blood cells, red blood cells, white blood cells, platelets don't escape out. Okay? Anything that's dissolved in the plasma can come out, but larger cells and plasma proteins do not come out. So now you can see this filtrate that comes out not only has waste products, it also has substances that are useful for your body, water and everything. So you can't lose everything that is filtered out. Okay? So that is where this tube comes in. As the filtrate passes through the tube, substances that are essential for the body are retained. They are taken back into the blood. Because it came from blood, they are taken back into the blood. And substances that are not needed remain in the filtrate so that they can be thrown out of the body. So that is why the tube is so important. And that's the reason why we don't call it urine at this stage, because it's not urine. There are lots of useful substances. Your body needs glucose, your body needs amino acids, it needs some of the salts. So all of that will be taken back, okay, back into the blood. And only the waste products will come out, whatever is not needed, water along with all the waste products will be thrown out. So this is what filtration is all about. So now you know, a healthy individual, if you examine the urine should never have blood and plasma proteins in it. Why? Because they don't get filtered. The nephron, the renal corpuscle is designed in a way that these things cannot come out. Okay, so you should not find plasma proteins, you should not find any blood in it, blood cells. If at all plasma proteins are found in urine or blood is found in urine, it does indicate that there could be a nephron damage, one of the reasons. Only if this is damaged, things can come out. Okay? Yes. So that is the composition of the filtrate. So your kidney is doing two things. The nephron is filtering the blood, and at the same time it is also taking back substances that are essential and only leaving behind in the filtrate whatever is not needed. Okay? So one more structure, let's talk about that, and then the second part would be the physiology. How is urine formed? So all of this you're going to be seeing in the models today. So one most important structure which Meredith was referring to earlier, the point at which the distal convoluted tubule, it kind of goes all the way up to the starting point of the nephron, and it comes very close to the afferent arteriole. It comes very close to the afferent arteriole. They almost touch each other, like you can see. Okay? This part where the afferent arteriole, the distal convoluted tubule meets the afferent arteriole or touches the afferent arteriole, this region is called the juxtaglomerular apparatus. Juxtaglomerular apparatus. What is it? It is the point at which the distal convoluted tubule comes very close to the afferent arteriole. Okay, and here there are a few things that are important. So we shorten it to say JGA, juxtaglomerular apparatus. Now, what does this this region, why is it so important? Because this is where you have sensors. You have sensors here. Okay. What are the sensors? You have sensors for blood oxygen levels. Okay. You have sensors for blood pressure. You also have sensors for blood sodium levels. You have sensors for all of these. Okay? And in response to the sense, once they sense low oxygen levels, it is cells in this region that also release erythropoietin, also release renin. So that's why this region is really important. It has sensors that can detect changes in blood oxygen levels, blood pressure, blood sodium, and at the same time, it can respond. The cells here can also secrete the hormones. Okay, so here's a magnified view of the same thing. So you can see the cells in the distal convoluted tubule, which is almost touching the afferent arteriole. They are modified, they are cuboidal cells, they are modified, they are called macula densa cells. They are called macula densa. And the afferent arteriole, the endothelium, that simple squamous epithelium which lines the blood vessel, okay, they are referred to as the juxtaglomerular cells. The afferent arteriole, the cells here, they are referred to as the juxtaglomerular cells, especially not just the endothelium but also the smooth muscle cells which are surrounding the arteriole. They are called the juxtaglomerular cells. So the macular densa cells along with the juxtaglomerular cells of the afferent arteriole are called juxtaglomerular apparatus. So it is right here that the sensors are present, and it is these cells that release erythropoietin and renin. So they sense as well as release. Okay? Yeah. So of course each of these cells and what they release, there's a lot of details, but as long as you remember, the juxtaglomerular apparatus is the region which has the sensors, and it also releases two hormones, renin and erythropoietin. Erythropoietin increases red blood cell numbers whenever oxygen levels are low. Renin, we know, is for blood pressure and blood volume. Renin, angiotensin, aldosterone mechanism. Okay? So that is about the overall anatomy part, okay, which normally we cover in the lab, but because I can't go straight into physiology without you knowing the structure of a nephron. So we covered it here. So hopefully the lab is going to be shorter. We'll do the anatomy, and then there's a little bit of physiology, urine analysis that we are going to do, okay, in the lab. So that's the thing. And now the next part that we will look at is the physiology. How is urine formed? Okay, that's the next one. Urine formation. So, blood supply is something that we have to look at. So maybe in the lab, if you look at the blood flow, we will take a look at this. So that's again identification basically. So not much to understand, but you will have to be knowing about all these blood vessels. And they are very clearly seen in a model. So we are going to be looking at the renal artery, what happens, how it divides, and how blood comes out of the renal vein. So we're going to look at the branches. Okay, so let's stop here. I'll see you in the lab at 2:30. We'll do the kidney, and please continue the same way. Like for those of you who came late, everybody did well on the second exam. The average was very good. So please keep it up and keep me happy.